Hello, I am Jitendrabhai Zaveri, and I am a sophomore at West Virginia University. I study computer science and neuroscience. My research interests are in machine learning and computer vision branches of computer science. Our research lab is called Vision Lab, and it's located in West Virginia University. I want to thank my mentor, my advisor, Dr. Gianfranco Doretto, the main contributor, Matthew Keaton, and other contributors, Cole Henderson and Meghana Kovur, for allowing me to present our research or project. Our project, it's called plant analysis. The end goal is to predict a particular plant species from a given example. And we are using fine-grained visual classification approach of machine learning. First of all, in order to understand this approach, we'll need to go through some basic machine learning introduction. So what is machine learning? As you can observe from its name, it's a system of algorithms that can learn automatically from its environment and also improve itself without explicit human intervention. So basically, it's an implementation of artificial intelligence. It can either be given past knowledge to predict future events, or it can also determine some patterns in given data set. For example, there are a lot of examples of machine learning algorithms, machine learning softwares in our everyday life. Google Translator, it's also a machine learning application. Self-driving cars, face recognition in your camera. So these are all machine learning approaches, machine learning algorithms. There are four primary methods, I would say most commonly used machine learning methods. First, supervised learning, unsupervised learning, semi-supervised learning, and reinforcement learning. So what is supervised learning? So we use this technique when data is given with labels. Basically, it uses past knowledge to predict future events. For example, if we want to predict an example of a dog, then we will train our algorithm using an image of a dog and a label attached to it. Furthermore, unsupervised learning. Unsupervised learning is used when the dataset is given without any labels. So it's basically unlabeled dataset or unclassified dataset. In order to perform this approach, algorithms will need to determine patterns in datasets. Semi-supervised learning. This learning falls between supervised and unsupervised machine learning. Usually, a small amount of data is labeled and large amount of data is unlabeled. Reinforcement learning. It's built upon the concept of awards and punishment, just like video games. This approach, we want to maximize the awards and minimize the punishment. So these were some most commonly used machine learning methods. We could also have a look at a few techniques, a few approaches in machine learning. Regression, which includes linear regression and logistic regression. Classification, which include binary classification, multi-class classification, multi-label classification. We are going to talk more about classification in upcoming slides. Clustering, anomaly detection, dimensionality reduction, assemble methods, neural networks in deep learning, transfer learning, neural language processing, and word embeddings. So in our approach, we are using classification method, and a label is attached with each training example. As I mentioned earlier, if we want to, if we want our model to learn to predict an image of a dog, then we will process a training example of dog with a label called dog. Similar goes for other examples. If you want to predict an image of a cat, we could do the same. We could use the same method or any other models. There are three most commonly used classification types: binary classification, multi-class classification, and multi-label classification. In binary classification, we use that when dataset only has two class labels. For example, we want to train our model which can predict an image of either of a dog or of a cat. So there are only two class labels available. So we will use binary classification technique here. Then there is multi-class classification technique. Here, we could use that when there are two or more class labels available in dataset. In our approach, we are using multi-class classification technique and multi-label classification technique. Here, each example might have multiple labels. So multiple labels can be predicted for each individual example. That's in multi-label classification technique. So in classification technique, we are using fine-grained visual classification method. So we use that when the categories for prediction are very similar, visually very similar. For example, if we want to recognize faces, then we could use this technique. Breeds of dogs, models of cars, bird species, and in our case, species of plants. We call they are usually subcategories. Let's have a look at some examples. Here, we're looking at a bird called cuckoo and species black-billed, mangrove, and yellow-billed. They look visually very similar. That's why, in order to distinguish each species, we will not be able to do that with normal machine learning approach, normal classification approach. That's why fine-grained visual classification will be the best choice. In our case, we want to predict plant species. Here, different plants have similar parts, similar organs, specifically leaves. In color, in shape, in size, they are very similar. That's why using fine-grained visual classification approach will be the best choice. Our project. It's one of the most popular challenges provided by PlantClef, which is also a part of ImageClef research initiative. They provide dataset each competition year. We're currently using dataset from the year 2015. This dataset is collected by untrained individuals. It means images often have a lot of background noise. It might also vary in quality and might also contain major occlusions. That's why we are using. That's why we are making bounding boxes around each individual organs to avoid background noise. We'll have a look at those in a bit. So the dataset contains 113,000 images, and which focuses on 1,000 herbs, trees, and fern species. And yeah, as I mentioned earlier, Each image will have bounding boxes, which are annotated by hand. Annotations are generated for the plant organs like leaves, flowers, fruits, stem, regions where the leaves are in high density, which means uh, if there is bush or whole tree. So this is our annotation code. We call it snappy annotator. Plant organs are annotated using different labels. Using their respected labels. So here, as you can see, a leaf is annotated with the label leaf. Here, I think the label is hidden for flower, but it's labeled flower. It's also other organs. Each bounding box's dimensions and their metadata is saved and exported into an XML file. So later use it when training. So each bounding box is kept compact to avoid background noise. As I mentioned earlier, there is a lot of background noise here. So here, when we annotate an organ, this leaf, it's not this another leaf is also in this part of bounding box. This we call background noise that we don't want. Also, we want to avoid overlap. Our approach is very simple, split in three parts. First, we want to develop an organ detector, which can detect a particular organ from an image. Here, we give an example. This image only contains leaves. That's why it's detecting leaves and processing it further. After detecting each organ, we are processing those detected annotated organs into their respected species classifier. So there will be 5 classifiers, species classifiers, which will then predict this particular species each organ belongs to. After predicting those species, a confidence score will be generated for each annotations, and then it will be the information will be fused together for the final prediction. Our approach is still in development. Currently, we are generating annotations in images. The dataset is huge. So once the annotations are done and they're processed to the algorithm, we'll have the results. Acknowledgements. I want to specially thank Dr. Stanislaw Pidorski for assisting us with the development of annotation tool Snappy Annotator. This work is based upon work supported by National Science Foundation, the grant number OAC 1761792. In machine learning, precision is usually dependent on the amount of dataset. We are performing annotations on the dataset, which contains over 113,000 images. But after it is annotated, we'll have more than that, because each image might also contain more than one organ. So our expectations for precision is very high. Our research lab. The research is published in conferences such as ICCV, CVPR, ICLR, ECCV. So we usually get 6 months of time span to finish our research, our project. If you guys have any questions, you can put it in comment box and I'll be really happy to answer you. And thank you for watching this presentation.